Hey guys, we're going to be doing a colony PCR using a master mix technique today. So that means that when I am setting up any sort of a reaction, and let's say I want to do a lot of the reactions, in this case 8, I have a couple options. I can either set up 8 reactions separately, or I can set up one big reaction in this tube and then just aliquot them out or squirt them out into my little tubes. So there's a couple of reasons why you would want to do a master mix. Number one, it's way easier. It saves you a lot of pipetting steps. And number two, probably a little bit more important, is that if you're going to be setting up each reaction individually, then you're probably increasing the likelihood that you're going to make a mistake, and pipetting error between all of those reactions is going to add up, so you have a bunch of slightly different conditions. Whereas if you make one big master mix, and then it's all the same in here, you aliquot it all out, you're guaranteed that every single one of those reactions is the same. That's kind of the motivation for the master mix. So then a colony PCR. That is a technique where we can take a colony and then just PCR that colony and get a sense for what we have inside of that colony. So here's a plate. I think the GFP is showing up. So we have a mixture of green and white colonies. So that's telling me I have mixed phenotypes here. And what I did was I attached a GFP on this half of this plate. I attached a GFP to a C1 gene. That's from lambdaphage. And so I have a bunch of green colonies, and then I know that, okay, those have GFP, but I don't know if they have that C1. So I'm going to be using a colony PCR to tell me, is the thing that is in my vector as big as GFP, or is it bigger than GFP? And if it's bigger than GFP, then I know I've attached my C1, and I know that assembly worked. The other way that we would do that would be to pick the colony, grow it up, mini prep it, digest it, and then run it on a gel. But we're just going to skip a day ahead, and this will allow us to screen through more colonies a little bit easier. So, on to the technique. So I have a flame, I have 2 sets of PCR strips. One of those sets is going to be for doing the PCR. The other is going to be to inoculate a little bit of culture. And then these 2 tubes are going to be kind of linked. So then I'm going to run my PCR, then I'm going to run it on a gel, I'm going to look at the bands, and then each one of those bands on the gel will correspond to one of these mini cultures, and depending on which one looks right, I will take this little mini culture and inoculate a big culture, and then I'll be much more confident actually that I am going to get the right construct. So we can start off with a PCR, because it's a little bit more complicated. So I have a little spreadsheet here, and I have the recipe for a single colony PCR, and then I put 9, so it multiplies everything by 9. That's my master mix formula. And even though I'm doing 8, I always add 1 extra, just because a little bit of pipetting error it can mess you up, and you want to make sure that you have enough for all of your tubes. So that's my recipe, and I guess we'll just start pipetting. So 189 microliters of water. I'm going to grab my P200, which is buried in here, and set it to 189. So we're going to be using boxed water, which is from a company that purifies the heck out of it, and it's really good reagent grade water. I'm going to go straight into my master mix tube, so 189 microliters of water. Then I want 22.5 microliters of tack buffer. So I'm actually going to use the same pipette. My P20 only goes up to 20. It's just out of range, so it's kind of on the edge of what this pipette can do, but it ought to work. So adjust all the way down to 22.5 and get a pipette tip. Find my tack buffer. Pull up 22.5 microliters. Inject that into my master mix tube and put it off to the side to tell myself that I've used it. Next, I want 4.5 microliters of DNTPs. So the DNTPs are the kind of the building blocks of the DNA, and these will get assembled by the polymerase in order to make that band that will run on the gel. So P20, 4.5 microliters. Here are my 2 millimolar DNTPs. Actually, I'm glad I double checked that. So that recipe calls for 10 millimolar DNTPs. I have 2 millimolar DNTPs. That could have been bad. So I'm going to put that over there. I'm going to go into my little reagent box here. I'm going to find another tube of DNTPs, 10 millimolars. Alright, that's what we want, 10 millimolar. 4.5 microliters, always double checking everything. Make sure you want to catch your mistakes. So now we have water, oops, we have buffer, we have DNTPs. A few more ingredients here. We want to add our primers. So we have 2 primers. In this case, our lab uses a forward primer called CA998. So 4.5 microliters of CA998 goes into that mix. And then our reverse primer, same volume, 4.5 microliters of G00101 into that mix. And then finally, we want 0.9 microliters of our polymerase, which is TAC. So I'm just going to run to the freezer and get that. But first I'm going to set my pipette to, I'm just going to do 1. So 1 microliter. Grab that enzyme. And it's not in here. Alright, here is our TAC. And this is the polymerase. I'm just going to make all the magic happen. It says 0.9, but I'm going to do 1. I'm feeling crazy today. So I'll pull up 1 microliter. I'm going to double check my pipette tip. Looks like it pulled up a little bit more than I wanted, so I'm going to kind of squirt it back down to the 1. Check that into my master mix. Put the enzymes back into the freezer so they don't warm up by being next to that flame. And then that is our master mix. So we have water, we have buffer, we have 2 primers, we have DNTPs, and we have our enzyme. So everything should be in there. We give it a quick buzz in the vortex. Tap it down. And now I'm going to aliquot it. So our aliquots are 25 microliters. So I'm going to grab my P200, set that to 25, click and lock it into place. Now I'm doing 25 exactly because I set up a master mix for 9. If I had set it up for 8, again, that pipetting error, what I could have done is just clicked it down a couple microliters and give myself a little bit of buffering. So 25 microliters is getting added into all of our PCR strips. 3, 4, 5, 6, 7, and 8. So now all those things are ready to go for PCR. But in order to do this colony PCR, we need to make sure that we are saving our colony, because as soon as we put our colony in there, well, not as soon as we put it in, but when we run this on the thermocycler, everything that's in there is going to die because it goes to really extreme temperatures, gets up to about 95 degrees. So I am going to get a little bit of LB with appropriate antibiotics, and that's why I've been having this flame on, because I want to have stereo technique. I'm going to flame that off, flame this off, and then I'm just going to start pipetting. So I'm actually going to put in about 100 microliters of media. 1, 2, 3, 4, 5, 6, 7, and 8. So now what we have is a Colony PCR master mix, and 8 of these PCR strips, and a bunch of LB in those PCR strips. And all I have to do is pick a colony, touch it to that PCR master mix, and then I'm just going to eject my tip in there and let the bacteria, the colony, just kind of soak into that mixture. Now I said earlier that I wanted to pick the green colonies, and it's a little bit hard to tell what's green or what's not. So I'm going to keep these glasses on, I'm going to shine the UV light on it, and get a little finicky, and I'm going to just pick the green ones, touch, touch. So, get a pipette tip, So look for a green colony. So there's a green one. I'm gonna touch it. You can see my pipette tip is a little bit green. I'm gonna give a quick touch and eject. I'm gonna repeat that over again. Look for a green colony. There's a green one. Pick it up. Touch. Eject. Green colony. Let's see. Sometimes they're really close to other colonies, so it's hard. Number 3. Touch. Eject. And another green one. 4. Touch. Eject. Halfway there. Another one here. There's a nice isolated one. Touch and up. Eject. 3 more. They're getting harder to pick out. There's one. Touch and eject. 2 more. That one. Touch, eject. Last one. How about, oh boy. I can't decide. How about that one? Touch, and then eject. Turn off my UV light, cap that plate. I'm gonna cap my PCRs, and then we're almost ready to go. I'm just gonna mix these things in, make sure that they're inoculated. Pretty much as soon as they touch, they're inoculated, but I like to get a lot of bacteria in there. Pulling out all these tips. Closing up this. And now the last thing is to just label it. So I'm just gonna do 1, 2, 3, 4, 5, 6, 7, 8. So 1, 2, 3, 4, 5, 6, 7, 8. Same thing on my culture tubes. 1, 2, 3, 4, 5, 6, 7, 8. And now we run our PCR. So we have the PCR, we have the culture. They're both labeled. We know that that tube corresponds to that tube, etc. So let's run on over to a PCR machine. And we can, looks like somebody else is done here, so we'll take this one over. And I know that the fragment in here is about 2 kb-ish. I'm actually going to run it as a 3 kb program just to be safe. I probably should have calculated that. I think it's actually a little bit over 2 kb, I can't remember. So the Taq polymerase, excuse me, runs at about 1 kb a minute. So that's why I want to make sure that I'm over the fragment lengths, otherwise it won't extend and you won't get banned. So I'm going to place my PCR mix into the thermocycler. I'm going to close it up. I'm going to run my program. We have a whole folder under Colony PCR. Call 3K, enter tubes, yes, 25 microliters, good, yes, 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 and it's going. So what it's going to do is it's going to ramp up to about 94 degrees for about 10 minutes. That's going to kill the bacteria, it's going to lyse everything, the DNA will survive. And then it's going to just start a regular thermocycling program. And eventually the parts will get amplified up and we'll be able to run them on a gel. So when that's done, we're going to run those on a gel, and the bands from left to right, 1, 2, 3, 4, 5, 6, 7, 8, are going to correspond to what is in these cultures. So what we'll do in the meantime is we'll just put this in the incubator and give these a chance to grow up. So those will slowly be getting a little bit more dense, and that's pretty much all there is to it. So we'll be back in a little bit, we'll run a gel, and then we'll just squirt those into some tubes, and then we'll know what we have.